Huntington disease strikes its terrible blow in adulthood, but genetic disorders that strike in childhood have an added awfulness, afflicting those who have hardly had a chance to live. Following a diagnosis, it is often possible to predict with grim certainty the course of the child's life. Such is the case with Duchenne muscular dystrophy, DMD, a progressive muscle wasting disease. DMD is a sex-linked disorder. The mutation responsible occurs in a gene carried on the X chromosome. Women may carry the mutation on one of their two X chromosomes, but they're usually protected by the presence of a normal version of the gene on their other X chromosome. It is highly unlikely a female will receive two defective copies, since males carrying the mutation almost never survive to have children. If, however, the chromosome with the mutated gene is passed to a son, the boy will develop DMD because he has no other X chromosome to supply a normal copy of the gene. When he is about 5 years old, his parents will notice he has difficulty getting up from the floor or climbing stairs. By about 10, he'll need a wheelchair. He will probably die in his late teens or early 20s. DMD is not rare. It affects 1 in every 5,000 male children. The hunt for genes involved in human disorders is a story dominated less by great research institutions and plucky entrepreneurs than by groups like the Hereditary Disease Foundation, organizations founded by those with first-hand experience of the devastation a particular genetic illness can bring. Led by people with something very precious at stake, these groups are by nature more willing to back risky or novel research, going where universities or biotech companies may fear to tread. The Muscular Dystrophy Association of America and its counterparts in Europe had long supported laboratory research directed at understanding the basic biology of Duchenne muscular dystrophy. In the late 70s, cytogeneticists, who study chromosomes microscopically, provided the first genetic clue. Among the very small number of girls who do develop DMD, an abnormality was found on the short arm of one of their X chromosomes at a location called XP21. Could this be the location of the DMD gene? Not long thereafter, Bob Williamson at St. Mary's Hospital Medical School in London initiated RFLP-based searches for both the gene causing cystic fibrosis and the one involved in DMD. His colleague, Kay Davies, hunted up RFLPs on the X chromosome and tested them for linkage to Duchenne. She was successful, and the clincher was their location. They were in the XP21 region, just as would have been expected given those strange X chromosomes in the women with DMD. While the gene hunters pushed ahead trying to isolate the genes involved in Huntington's disease and DMD, a revolution of a quieter kind was taking place in the offices of clinical geneticists. From the first, Nancy Wexler and David Hausman realized that RFLPs linked to a disease gene could be used not only to localize the gene itself, but also as a diagnostic test to determine which members of a particular family were carrying the mutation. They could be used to test even an unborn child. Consider the case of a hypothetical family with DMD. At least one boy will be diagnosed, the index case that first reveals the presence of a DMD mutation in the family. His mother, the carrier of a mutated gene, also has one normal copy. Her sisters may also be carriers, so any sons they may have are at risk. Now suppose the mother becomes pregnant once again with a male fetus. The chances are 50-50 that the second son will be affected. But with RFLPs, her physician can tell her what fate awaits that fetus if carried to term. First, the affected son's X chromosome is analyzed to identify the particular RFLPs linked to the DMD gene in this family. Next, DNA is taken from the fetus. either a sample of the placenta or of the amniotic fluid, which contains fetal cells. If the fetus's RFLPs match those of the affected boy, then we can be pretty certain that the unborn fetus will also be affected. Why only pretty certain? As we saw in Chapter 1, when egg cells are produced, the chromosome pairs undergo recombination, exchanging DNA. The two copies of chromosome 1 trade with each other, as do the two copies of chromosome 2, the two copies of the X chromosome, and so on. If this swap should occur at a point on the X chromosome between the RFLP markers and the DMD gene, the RFLPs we have found to be associated with the normal version could possibly wind up associated with the mutated DMD copy. Experience taught us that with the first RFLPs for DMD, this happens about 5% of the time. And so RFLP-based diagnosis has only a 95% chance of being accurate. This degree of imprecision is an unavoidable consequence of recombination. So, while such diagnosis represented a tremendous advance, absolute certainty depended on identifying the gene itself, not merely the markers associated with it. The key to isolating the DMD gene was a young boy named Bruce Breyer, whose X chromosome was missing a very large piece from the short arm. The piece was so large that Bruce suffered from 3 other genetic disorders, in addition to DMD. In 1985, Lou Kunkel at Harvard Medical School reasoned he could use Bruce's DNA to fish out a normal gene from the DNA of an unaffected boy. Bruce's case was special because the disease was caused not by a defective copy of the gene, but by its complete absence. Kunkel realized that all of Bruce's DNA should be present in a normal boy's. But whatever sequences the latter had, and Bruce lacked, would hold the key. Using recombinant methods, Kunkel subtracted Breyer's DNA from the normal DNA and kept the difference, the DNA that should contain the DMD gene. The subtraction didn't work perfectly, but it did work well enough that he could find the DNA pieces he wanted by using genetic markers associated with the XP21 region. Tony Monaco, a graduate student with Kunkel, took on the job of determining which, if any, of these XP21 pieces of DNA might constitute part of the DMD gene itself. The only way to do this was to test each piece against DNA from several unrelated patients with DMD. Monaco hit the jackpot with the 8th try. A sequence called PERT87 was found to be absent in 5 of his DMD boys. This meant almost certainly that PERT87 was very close to the gene, and perhaps even a part of it. Monaco began to isolate other sequences close to PERT87, and these too proved to be missing in the DNA of DMD patients.